Sea Urchin Fertilization by Joe, Alexa, Mara, Casey, and Nathan, Group 33. Fertilization
is responsible for the continuation and evolution of all organisms. To study fertilization, researchers often study sea urchins because they have many easily testable features. Sea urchins offer large quantities of sperm and egg cells for studies. Also, during fertilization, the sperm and egg meet and interact outside of the organism's bodies, allowing researchers to follow the process in the seawater environment.
Sperm are attracted by chemotaxis, which is a gradient of chemicals secreted by the egg that allow for the sperm to find the egg. One example of a chemotactic molecule is a 14 amino acid peptide called Resac, which has been isolated from the egg jelly of a sea urchin. Resac diffuses readily in seawater and has a large effect at very low concentrations. When added to sperm suspensions, the sperm tend to congregate around the Resac. Resac is specific and does not attract sperm of any other species. Sperm have receptors in their plasma membrane that bind to Resac and can swim up a concentration gradient to the egg. Resac also acts as a sperm-activating peptide in which it dramatically increases mitochondrial respiration and sperm motility. The receptor for Resac is a transmembrane protein which binds to Resac on the extracellular side. Conformational change on the cytoplasmic side activates the receptor's enzymatic activity, which in turn activates a mitochondrial ATP generating apparatus which stimulates the flagellar movement in the sperm. The second interaction between sperm and egg is the acrosomal interaction. There are two components of this. These are the fusion of the acrosomal vesicle with the sperm plasma membrane and the extension of the acrosomal process. Acrosomal reaction in sea urchins is initiated by contact of sperm with the egg jelly. This contact causes the exocytosis of sperm's acrosomal vesicle and the release of the proteolytic enzymes that digest the path through the jelly coat to the egg surface. It is thought to be initiated by a fucose-containing polysaccharide in the egg jelly that binds the sperm and allows calcium to enter the sperm head. Exocytosis of the acrosomal vesicle caused by calcium-mediated fusion of acrosomal membrane with the adjacent sperm plasma membrane. Extension arises through polymerization of globular actin molecules into actin filaments.
To gain proper sperm cell to egg cell specificity, the sperm cell is coated with a protein called bindin. Bindin can be found on the sperm acrosome, an organelle that develops over the anterior of the head in the sperm cells. This bindin acts like a glue between the sperm cell and the egg cell. However, once isolated, bindin shows an affinity for a carbohydrate on the egg cell surface, thus providing the sperm with a target receptor. Bindin controls the fusion of the two gametes. Depending on the liposomes in the egg cell, the bindin will either aggregate vesicles or fuse them. In gel phase vesicles, bindin aggregates the vesicles, while in mixed phase vesicles, bindin fully fuses them. The fusion in each case can be increased by the introduction of zinc, which changes the conformation of the bindin core, also known as B18, and allows for more fusion.
Sea Urchin Fertilization. Sea Urchin Fertilization